The Swedish histochemists published the maps of where the monoaminergic neurons were. The study with LSD and the serotonergic neurons, that was the first recordings from identified monoaminergic neurons. And then within the next 2 or 3 years in my lab, we went on to record from the dopaminergic neurons and the noradrenergic neurons of the locus ceruleus. So that series of studies, they were the first recordings from monoaminergic neurons, and the first studies which describe their basic electrophysiological properties and the effects of drugs on their firing rate. That started off, it's become a major industry. In Britain, there were those that were inclined toward thinking that serotonin would have a role in affective disorders. In Sweden, Arvid Carlsson believed that serotonin may be important for affective disorders or depression. Arvid Carlsson was involved in the development of serotonin reuptake inhibitor zymilidine, but it had certain toxicity and was dropped. Meanwhile, Eli Lilly was developing fluoxetine and Prozac, and they were the first to market on that. But actually the basic science work had gone on for some years before Prozac ever got into clinical testing. Atypical antipsychotic drugs that we've had in the past also have other actions. It's difficult to sort out the 5-HT2A component from the other components. Risperidone, I think, comes the closest in the currently accepted drugs of a drug that has very high potency at blocking 2A receptors. It's about 10 times more effective at blocking 2A receptors than, let's say, D2 receptors. Risperidone has now been clearly shown to be a completely efficacious antipsychotic. But now there is a drug. It was called MDL-10907. Now it's called M-10907 because there was a corporate takeover. That's just gone through phase 3 clinical trials. Quite interesting that ritanserin doesn't seem to do the trick, even in animal models. One hears various rumors that it works. The question in my mind is, does it work in just a subset of patients? Being so selective, maybe we all believe that probably schizophrenias are not all the same. Might it pick out a subset of schizophrenias where the 2A receptor is important in the pathogenesis? It's an interesting coincidence that these receptors are concentrated in the cerebral cortex. In recent years, we've been studying electrophysiologically what the role of 2A receptors. And what we've found and have reported is that 5-HT2A receptors are activated in the cortex. They increase glutamate synaptic potentials. They increase excitatory postsynaptic potentials. They increase it very dramatically. Serotonin does it, hallucinogens do it. Hallucinogens are partial agonists of 2A receptor, so they actually don't work as well as serotonin. What makes the hallucinogens, we believe, is that the other actions of serotonin counterbalance the effect on increasing glutamate transmission, whereas with the hallucinogens, there's no counterbalance. So the 2A effect is unopposed. That's our belief at this time. So we've proposed the idea that it's not necessarily an overactivity at 5-HT2A receptors that might be responsible for endogenous states, but an abnormality in glutamate release that underlies this. Because if it was a problem in excessive 2A receptor function, drugs that block 2A receptors should work right away, and they don't. Other studies have been going on with the ketamine model of psychosis. Ketamine being a non-competitive antagonist of one type of glutamate receptor, the NMDA receptor. And the ketamine model, for many, many years, has seemed a completely separate, different model than the psychedelic hallucinogen model. Recently, it's been found by Beta Mogadam, who is a member of ACMP, that ketamine induces an increase in glutamate release in the cortex, so that the ketamine model may be a model that's based on increased glutamate release. But that's exactly what we're saying about psychedelic hallucinogens, that there's an increase in glutamate release. Now, on top of this, there's PET imaging studies, which in Europe currently investigators are able to give psychedelic hallucinogens. I don't think that's true here, but there's a laboratory of Vollenweider where they give them mescaline and psilocybin. And what they've shown is hyperfrontality, which they think corresponds to what happens in acute states of schizophrenia. In that same laboratory, and also in other laboratories, it's been reported that ketamine induces hyperfrontality. I think now the new basic studies could account for that, because in both cases you get an increase in glutamate release. So now we're proposing that there's a commonality to the psychedelic hallucinogen and ketamine models of psychosis, and that is increased glutamate release. One of the drug companies associated with ACMP over the years, Eli Lilly, the Prozac company, they're the leading developer of metabotropic receptor agonist drugs. These drugs are rigid analogs with different side groups of glutamate itself, and it was believed that such drugs could never enter the brain because they were too polar. But they've developed very highly potent agonists, nanomolar potency agonists, that can be given systemically and enter the brain. These drugs were originally developed for use in treatment of anxiety disorders, and these drugs are being used in clinical trials. But now, because of this new data implicating excessive glutamate release, Eli Lilly, through our efforts and Beta Mogadam's efforts, are now contemplating clinical trials in schizophrenia.